Welcome to The Explainer. Today, we're diving into a really fascinating intersection of chemistry, biology, and artificial intelligence. We are going to unpack a brilliant review article called Structure-Based Drug Design with Geometric Deep Learning, put together by researchers Clemens Isert, Kenneth, and Gisbert Schneider. If you've ever found yourself wondering how we use cutting-edge computer science to actually discover and design entirely new medicines from scratch, well, you are in exactly the right place. Let's get into it.
So here is the big question that pretty much drives this entire field. Can artificial intelligence actually learn to navigate the three-dimensional world of molecules? You see, for a really long time, we've drawn molecules as flat, two-dimensional shapes on a piece of paper or a whiteboard. But obviously, that's not reality. Biology happens in full 3D space. So how do we go about teaching an AI to understand a non-flat, highly complex, and constantly shifting 3D molecular world?
Okay, let's dive into this with a classic analogy, but we're going to upgrade it a bit to really grasp structure-based drug design. Think of the biological lock and key. Imagine a protein in your body as a microscopic biological machine. A drug molecule is essentially a tiny key trying to interact with that machine to change how it works. But here's the catch. This isn't your standard flat, rigid metal lock. It's a complex, squishy, flexible 3D object made entirely of atoms, tiny pockets, and moving parts. The drug literally has to fit perfectly into this shifting 3D landscape to do its job.
Section 1. The 3D Drug Puzzle.
To actually solve this puzzle, we have to understand why three-dimensional space is the fundamental challenge and really the ultimate key to discovering new drugs. We rely heavily on what's called structure-based drug design. Basically, this means we use the actual 3D geometry of a target, like one of those proteins, to design a molecule that specifically binds to it. It's simply not enough to just know the chemical formula of a potential drug. We have to predict exactly where it binds, estimate how strong that binding affinity is, and figure out its exact 3D pose, which just means its orientation and shape once it's tucked inside that protein pocket. Ultimately, understanding a drug's effectiveness is entirely dependent on understanding its physical geometry in 3D space.
Section 2. Why standard AI fails.
Now, you might be wondering, why can't we just use ChatGPT or standard AI image generators to figure this out? Well, standard neural networks just weren't built for biology. It fundamentally comes down to the difference between Euclidean and non-Euclidean space. Standard AI, like the models that instantly recognize your face in a photo, thrives on Euclidean data. That means regular, flat, predictable grids, kind of like pixels in an image or squares on a chessboard. But molecules, they are non-Euclidean. They're wildly irregular. An atom of carbon might connect to 4 other atoms, while an oxygen atom nearby only connects to 2. There's no fixed left, right, up, down grid. It's just a tangled friendship network of atoms. Because of that, ordinary deep learning gets completely lost trying to process this irregular structure.
Section 3. Teaching AI 3D Geometry.
And this brings us to the real hero of our explainer, geometric deep learning. This is a highly specialized form of machine learning designed from the ground up to handle those messy, non-Euclidean structures. To actually use it, we first have to translate a protein into a format the AI can see. The source review highlights 3 main ways we do this. First up, 3D grids, which slice up space into little 3D cubes called voxels. Second, we have 3D surfaces, which map out the outer boundary of the protein, almost like throwing a polygon mesh over it. And third, 3D graphs. These treat the molecule as a network of nodes and edges, which perfectly mirrors the actual atoms and chemical bonds. Now, what's really interesting about this slide is seeing all 3 of these methods applied to the exact same target. What we're looking at is the human MDM2 protein, with a specific drug ligand known as DF1 docked right inside it. On the left, you've got that voxel grid representation. It honestly looks a bit like a Minecraft build, right? Slicing the space up into tiny cubes. In the middle, the mesh surface beautifully captures those physical pockets and how the shapes complement each other. And on the far right, the 3D atomic graph maps out the precise interconnected relationships between the atoms themselves. Each method gives the AI a totally unique geometric perspective on the exact same protein.
Section 4. The Rules of Symmetry.
Handing an AI a 3D model is a huge step, but it's not quite enough. The AI also has to intuitively understand the physical rules of how these objects exist and move in real space. So let's do a quick mental checkpoint here. Imagine you have a protein-ligand complex, and you predict its binding affinity, basically how tightly the drug holds on. If you take that exact same 3D complex and simply rotate it upside down in space, should the predicted binding affinity change?
The answer obviously is no way.
The physical molecule hasn't magically changed just because we turned it around to look at it from a different angle. This introduces an absolutely crucial concept called invariance. A good geometric deep learning model must be invariant to certain transformations. Meaning, if the input rotates, translates, or shifts, the scalar output, like that single number representing binding affinity, must remain exactly the same.
But hold on, what if the AI isn't trying to predict a simple scalar number, but a direction in space, like a force vector? Well, that requires something called equivariance. If the input changes, the output must change in a completely predictable, deterministic way. Think of it like this. Imagine a little toy car with an arrow glued to its roof pointing forward. If you physically rotate that car 90 degrees, that arrow must also rotate exactly 90 degrees with it. That right there is equivariance, and it's essential for teaching an AI how real 3D forces operate in biology. And this brilliantly illustrates exactly how those rules play out. Looking at the symmetry groups mapped out here, E3 covers translations, rotations, and reflections. SE3 is similar. It covers translations and rotations, but it intentionally ignores reflections. Why? Because in biology, a mirror image of a molecule might be highly toxic, even if the original version is completely safe. It's a massive distinction. And below that, you can clearly see how an equivariant model actively rotates the output factor right along with the input molecule, while an invariant model just stubbornly outputs the exact same scalar number, like 4.73, regardless of how you spin the molecule.
Section 5. Designing drugs from scratch.
Okay, so once an AI genuinely understands these 3D geometries and physical symmetries, we can make a massive leap from just analyzing existing molecules to actually creating brand new ones. Historically, finding a drug's 3D pose, a process called docking, treated the protein like a totally rigid, inflexible block. It was slow and frankly pretty limited. But geometric deep learning is a real game changer here. Models like DiffDock use a generative diffusion process. Imagine starting with a drug molecule just floating randomly in space. Step by step, the AI basically acts like it's denoising a blurry image, gradually shifting, rotating, and tweaking the molecule's tiny joints until it perfectly snaps into a highly plausible binding pose. And going even further, we have de novo design. This is where the AI doesn't just dock a molecule we already know about, it literally invents a brand new one from scratch, atom by atom.
Let's move on and see how this actually builds into a functional design pipeline. The AI first looks at the 3D surface of a protein's binding pocket. It then mathematically encodes that complex 3D shape into a tightly compressed sequence of numbers, known as a 1D latent space. Once it does that, a chemical language model kicks in and acts as a decoder. It reads those numbers and spits out a text string called a SMILE string. This text string represents the exact chemical formula of a brand new drug designed specifically for that pocket, just like the DF1 molecule we looked at earlier. It's essentially translating a physical 3D shape directly into a chemical recipe. Pretty mind-blowing, right?
Now, as incredible as all of this sounds, we definitely need a reality check here. The authors of the review are incredibly clear about the current limitations. First off, data quality. An AI is only as good as the data you feed it, and high-quality, real-world 3D experimental structures are still somewhat limited. Second, we have this issue of memorization versus learning. Some models don't actually learn the underlying physics. They just memorize their training datasets, like PDB bind. It's like a student memorizing an answer key without actually understanding the math. Third, flexibility. As we mentioned, proteins are squishy and they move, which makes flexible docking incredibly hard to simulate accurately. And finally, lab testing. At the end of the day, an AI prediction is just a hypothesis. Until that molecule is physically synthesized in a lab by humans and tested in real biology, it remains nothing more than a very, very educated guess on a computer screen. So, as we wrap up this explainer, I want to leave you with this final thought to ponder. We are currently shifting from a decades-long era of drawing 2D chemical formulas on flat paper to a new era where we must fundamentally teach machines to perceive and manipulate the complex, folding, non-Euclidean geometry of life itself. Does the future of medicine rely less on the traditional molecules we already know, and more on how well we can teach artificial intelligence to truly see in 3 dimensions? It's an absolutely fascinating frontier, and geometric deep learning is leading the charge. Thanks so much for joining me on this deep dive into structure-based drug design. Catch you next time.